Hi, and welcome to Mr. Fisher's Biology Podcast series. This podcast is entitled The Scientific Method and deals with how scientists do science. Before we begin, what I want you to do is pause the podcast and simply just copy down this flowchart in your notes. This flowchart is a very good flowchart that goes over the basics to the scientific method and would be very useful in lab situations. Okay, so looking at the scientific method, good science always starts with good observations. So observations about the environment or anything in the natural world using our five senses. Observations usually follow, or following observations are usually questions. Questions are based on observations, and we can generate one or more questions based on that. So looking at our little dinosaur example here, a paleontologist noticed some rocks, noticed some fossil formations. A logical question would be, what kind of dinosaurs do these bones come from? The next step is to form a hypothesis, and we'll look at the definition in just a second. But a hypothesis, or a statement that we, a question that we ask, is testable if we can collect evidence to either support it or not. Hypotheses are never proven beyond a doubt, and in a lot of cases they need to be refined, revised, or a lot of them are just discarded. So going to our paleontologist, these bones come from a brand new dinosaur. That's a pretty logical hypothesis for that question. So a hypothesis is a statement. It's made in advance. It states the results that will be obtained from testing the hypothesis. A lot of students have trouble when it comes to creating a hypothesis, and this is why I would like you to follow this format. If you follow this format, which I call if-then statements, you'll have a very good logical hypothesis. So for example, if we change light intensity, then plant growth will increase. So in this example, or in this hypothesis, it's really something that you can see us testing. The statement here, the if statement, is what we're going to be changing in our experiment, and we'll go over what we define as what we change. And then what we sort of look at in our experiment is part of the then statement. So if we're going to change the light intensity, and you can imagine how you could do that, then you can imagine what the outcome would be. So when you're creating your hypotheses, try using if-then statements. And in a lot of cases, for a lot of different experiments, this will be the best method. Step 3. Step 3 in an experiment is the experiment. So we test our hypothesis, basically, and we do that by gathering data, and we gather data under controlled conditions. So we call this conducting a controlled experiment. And what that means is that we're changing something in the experiment, but we're keeping everything else the same. So what we do is we base our experiment, which we call usually our experimental group, on some sort of a control group. So going back to our light intensity, we may have one set of plants that are growing under normal light conditions. And we have plants that are growing under different light intensities. And we're going to compare the growth based on those different light intensities. So that's an example. In an experiment, both groups, so our control and our experimental group, are identical except for one factor. And this is what we call the independent variable in an experiment. I want you to remember independent variable like this. What you change. So what you change in an experiment is your independent variable. Now, When conducting the experiment, we take a series of observations and measurements, and that's our dependent variable. But how I want you to remember it is what you measure. So what you change in an experiment is your independent variable, and what you measure in that experiment, that's your dependent variable. So your dependent variable is driven by or results from the independent variable. So here's an example. This is a good biology example. This is a good title, testing the effect of UV light on frogs. So what you can see is that there's two experimental groups that are present within this experiment, and there's a control group. And remember, control group is sort of normal conditions. Experimental groups were changing something. So if we look at the experiment, we notice that the frogs are the same. So that's a control. The number of eggs that we use in this experiment, that's the same. That's a control. The water temperature was kept the same. So that's a control. But here's what we changed. So in the experiment, we changed the UV light exposure. Now in our control, we didn't expose the eggs to any UV light whatsoever. And in experimental group 2, we exposed them over 15 days, and experimental group 3, we exposed them over 24 days. So this is our independent variable, the UV light exposure. And our dependent variable is a little bit more difficult. Our dependent variable we're going to be looking at, because we're looking at eggs, we're probably going to be looking at the hatch rate, I would say, in this experiment. So in this case, the dependent variable is not that clear, but we can gather it from the data table itself. So when it comes down to measuring, we have what's called quantitative data. That's numbers, so weights, heights. And then we have qualitative data, and those are things that aren't numbers. So things like smell, taste, those are qualitative. Sampling is actually a technique that uses a small part to represent an entire population. So obviously, if we look at maybe an environment, if we're measuring the population of animals, it would be very hard for us to take all of the animals and measure that population. I mean, it would just be too big. And so oftentimes what a biologist will do is we'll take a section of animals, and that will represent the entire population. Step 4. Once data is collected, it needs to be organized. Data is organized in the following formats: graphs, charts, tables, and in some rare cases, maps. In this biology course, you'll be using a lot of graphs. You'll be putting a lot of tables together. You may not ever see a chart, and you will not see maps. But they're very common ways for us to organize data. Again, here's an example of how we can take data in this information, and in this case, they constructed a line graph to display that information. Once we organize data, we need to analyze data. So this is what we look at our data that we have present, and we go back to our hypothesis, and we say, does the data that we collect support the hypothesis that we stated? If it does, then we can make a conclusion. If it doesn't, we can still make a conclusion. And in a lot of cases, we may have to go back and, remember, refine or even discard our hypotheses. Step 5 is conclusion. Conclusions are made on the basis of facts, not observations. So we look at the data that we collected and we make a conclusion based on the facts. We draw it from our experiment. It should support the hypothesis, and in a lot of cases, it should also be retestable. Step six, obviously, once we've conducted our experiment, reached our conclusions, a good scientist needs to be able to communicate it. And there's a variety of methods that communication is done, published in journals, scientific meetings. One important point to note is that with science, scientists must be unbiased. So data should not be tampered with, and we should only publish and report tested and proven ideas. Okay, so science is an unbiased way of explaining the natural world. Sharing information is essential, and oftentimes information is subjected to more examination and verification, so scientists will actually retest the hypothesis multiple times in order to verify that it is accurate. And this is how we get truth to science. Which brings us to two important ideas, the ideas of theory and laws. And these are often misrepresented when it comes to definitions. So theories may be formed after hypotheses have been tested and supported with experimental evidence. A theory is the following: a broad and comprehensive statement of what is thought to be true. Theories are supported by considerable evidence, and theories tie together related hypotheses. So for example, the theory of evolution is a broad idea. It's a broad statement of how organisms change over time. It incorporates many hypotheses, and it's got hundreds and hundreds and thousands of experiments that have been done and tested on it. And it's what we believe to be true of the natural world. Laws are a little bit different. A law is a statement of fact that explains an action or a group of actions. So for example, gravity. We know that when objects fall, they fall down. That's the law of gravity. It's accepted to be true. So there really isn't a person on the planet that would argue that on Earth objects fall down. It's universal. And that can be a little bit deceiving. It's universal to the specifics of the law, the confines that the law says. So the law of gravity works in this solar system, but it's applied to the planet Earth. And oftentimes laws can be expressed as mathematical equations. So the law of relativity equals mc squared. If you see a math statement behind it, generally speaking, you're looking at a law. That concludes the Scientific Method podcast. Thank you.